Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study on polioencephalopathy in Eurasier dogs, a topic that sheds light on the genetic complexities underlying neurological disorders in canines. This research, spearheaded by Faye Rawson and colleagues, explores the clinical and genetic characteristics of this condition in a family of Eurasier dogs, offering valuable insights into the hereditary nature and potential genetic causes of polioencephalopathy. Polioencephalopathy, or P, is a condition marked by multifocal, bilateral, and symmetrical lesions in the gray matter of the brain. These lesions often arise from metabolic disturbances, such as nutritional deficiencies or inborn errors of metabolism. In human medicine, mitochondrial disease is increasingly recognized as a cause of PE, leading to a range of clinical signs, including cognitive regression, impaired motor function, and movement disorders. The challenge lies in the heterogeneity of clinical presentations, making genetic testing crucial for early and accurate identification. In veterinary medicine, genetic investigations have expanded our understanding of uncommon encephalopathies, but only a few causative genetic variants for mitochondrial encephalopathies have been identified in dogs. This study focuses on 3 young, related Eurasier dogs affected by PE with atypical manifestations, characterized by the early onset of movement disorder episodes, progressive impairment of motor function, and preserved consciousness and behavior. The investigation suggests MECR to G as a possible causative variant for this presumed hereditary and progressive PE in these littermates, sharing similarities with a rare form of childhood-onset dystonia reported in individuals with MECR loss of function variants. The study began with ethical approval from the University of Bristol Animal Welfare and Ethical Review Body and the Cantonal Committee for Animal Experiments. Blood samples were collected from 2 male Eurasier littermates, dogs 1 and 2, who presented with movement disorder episodes since 9 weeks of age, and from a female littermate, dog 3, with similar but milder exercise-induced episodes at 4 to 6 months. Additional samples were taken from 3 healthy littermates, both parents, and 115 Eurasier dogs from the VetSuisse biobank as a control group. Genetic analyses involved extracting DNA from blood samples and genotyping the entire family on Illumina canine bead chips. Homozygosity mapping and parametric linkage analysis were performed, assuming an autosomal recessive inheritance model. Whole genome sequencing of dog 1 was conducted, and the sequence data were deposited in the European Nucleotide Archive. The ATG4DC to T and MECR to G variants were genotyped in the family and the control group via Sanger sequencing of PCR amplicons. The clinical descriptions of the affected dogs reveal a complex and progressive neurological condition. Dogs 1 and 2 were presented to UK-based referral centers for recurrent neurological signs. Dog 1, a 21-week-old male, had a 12-week history of recurrent movement disorder episodes, initially infrequent and coinciding with overstimulation and loud noises. These episodes were characterized by generalized ataxia, a hypometric thoracic limb gait, dystonia, and irregular flexion and extension movements of the thoracic limbs. As the condition progressed, the episodes increased in frequency and severity, leading to marked exercise limitation. A divergent ventrolateral strabismus of the left eye and decreased peripheral vision were also detected. Dog 2, a 15-week-old male, had a 6-week history of abnormal movement disorder episodes, initially exercise-induced, with intermittent hopping on the thoracic limbs and collapsing on the pelvic limbs. Later, a head tilt, strabismus, and increased tone in the thoracic limbs were observed. The episodes increased in frequency, resulting in moderate exercise limitation. Dog 3, a female puppy, experienced movement disorder episodes when exercised for more than 20 minutes. Video footage showed increased tone in all 4 limbs, more pronounced in the thoracic limbs, leading to abnormal posture and gait. Exercise restrictions substantially decreased the frequency of these episodes. Clinical investigations in dogs 1 and 2 included complete blood counts and serum biochemistry, which were mostly unremarkable. Both dogs had normal bile acid stimulation test results, while dog 1 had a mildly increased ammonia concentration. Both also had mildly increased CK activity and normal AST activity. Tests for toxoplasma and neospora were negative, and cobalamin, folate, and thiamine serum concentrations were normal. Urine organic acids analysis in dog 1 was comparable to a healthy control dog. Electrodiagnostic studies in dog 2 were normal, with normal electromyography and nerve conduction velocities. Cisternal cerebrospinal fluid (CSF) analysis was also normal, and PCR tests for toxoplasma and neospora in the CSF were negative. Magnetic resonance imaging, MRI, revealed symmetrical, bilateral multifocal T2 and fluid attenuated inversion recovery, FLAIR, hyperintense, T1 hypo, to isointense, non-contrast enhancing lesions throughout the gray matter of the caudate nucleus, lateral and medial geniculate nuclei, thalamus, hippocampus, and rostral colliculus. Dog 1 had more severe lesions, particularly in the thalamus and hippocampus, while Dog 2 showed more involvement of the rostral colliculus. Treatment for dogs 1 and 2 included clonazepam, which decreased episode frequency. Thiamine supplementation showed no response, and carnitine administration in dog 1 led to transient deterioration. Anti-inflammatory doses of glucocorticoids and chlorphenamine were also ineffective. Both dogs were fed Purina Pro Plan and C Neurocare, and Dog 2 was treated with Activate, but it was discontinued due to a lack of response. At a 1-year follow-up, Dog 1 showed generalized muscle atrophy. Neurological examination revealed difficulty rising and walking, a hypometric gait, and bilateral divergent ventrolateral strabismus. Spinal palpation was unremarkable. By the time of writing, Dog 1 was 26 months old, with mild disease progression managed by restricted exercise and clonazepam. Dog 2 was euthanized at 25 months due to a prolonged movement disorder episode. Dog 3, at 15 months, appeared well-controlled with exercise restriction. The genetic analyses focused on identifying a monogenic autosomal recessive mode of inheritance, considering that the phenotype in Dog 3 was milder than in Dogs 1 and 2. Combined linkage and homozygosity mapping identified several intervals spanning 103 megabits when only Dogs 1 and 2 were classified as affected, and 84 megabits when Dogs 1 to 3 were classified as affected. Whole genome sequencing of Dog 1 identified a single private homozygous variant with likely functional impact in each of the critical intervals. An ATG4D variant was identified in the critical interval derived from Dogs 1 and 2, while a MECR variant was found in the critical intervals resulting from the analysis, including Dogs 1 to 3. Both variants are predicted to lead to an exchange of a single amino acid in the encoded proteins. Genotyping of the entire family showed that the genotypes co-segregated as expected with the severe ATG4D and MECR, or mild neurological phenotype, MECR only. Neither mutant allele was present in 115 additional genotyped Eurasier dogs without similar neurologic disease. The study identified a homozygous missense variant in MECR as a possible candidate causative variant for a previously unreported presumed hereditary and progressive PE in the 3 Eurasier littermates. An additional homozygous missense variant in ATG4D was identified in the 2 most severely affected dogs, suggesting potential additive effects. The MECR gene encodes mitochondrial trans-2 enoyl coenzyme reductase, a protein involved in the synthesis of lipoic acid, LA, in eukaryotic cells. Lipoic acid is essential for mitochondrial fatty acid synthesis. Inborn errors of metabolism affecting LA synthesis resemble typical mitochondrial disorders, involving organs with high energy demands and susceptibility to oxidative stress, such as the basal ganglia and optic nerves. Specific disruption of the MECR gene caused neurodegenerative changes and Purkinje cell loss in a mouse model, supporting the gene's crucial role in cellular metabolism and neuronal survival. Clinical signs and radiological findings in the 3 dogs share similarities with those reported in people with MECR variants and childhood-onset dystonia and optic atrophy. Children with this rare, progressive, and debilitating neurodegenerative disease show early dystonia, decreased balance and motor function, vision loss, and dysarthria. MRI identifies bilateral hyperintense T2W signal in the caudate and putamen nuclei. While clinical and MRI findings overlap with other mitochondrial encephalopathies, this condition differs by the relative sparing of cognition and absence of additional organ involvement and typical mitochondrial biomarkers. The clinical presentation in the 3 dogs reflects simultaneous involvement of multiple neuronal systems with movement disorder, dystonia/Bailism, and progression of cerebellar signs, but no cognitive decline or behavioral changes. The dogs also lack systemic signs or laboratory changes suggestive of multi-organ failure. Contrary to reports in humans with childhood-onset dystonia and optic atrophy was not identified in any of the dogs. The absence of optic nerve involvement may be due to the early stage of disease in these dogs, but it is not entirely atypical for individuals with MECR variants. Recent research suggests replacing the term childhood-onset dystonia and optic atrophy with mitochondrial enoyl CoA reductase protein-associated neurodegeneration (MEPN) due to the heterogeneity of clinical phenotypes. Currently, there is no specific curative treatment for encephalopathies not associated with nutritional deficiency. Dietary supplementation with LA or its precursor, octanoic acid, is recommended in people with MECR variants to halt or slow disease progression. LA supplementation did not appear to modify the course of the disease in one Eurasier dog, suggesting potential differences in biochemical pathway disruption with the mutated MECR gene between species. Additional treatment trials in dogs 1 and 2 targeted GABAergic, clonazepam, and cholinergic, chlorpheniramine, neurotransmitter systems. Benzodiazepines have been used previously in dogs with paroxysmal hypertonicity disorder and in humans with akathisia. While chlorpheniramine showed no response, the promising response to benzodiazepines could warrant further trials with antiepileptic medications. Dopaminergic treatment, such as L-Dopa, was not used in these dogs. A further homozygous MECR variant in ATG4D was identified in dogs 1 and 2. The ATG4D gene encodes a cysteine protease belonging to the macroautophagy pathway. ATG4D variations disrupt neuronal homeostasis, particularly in axons and Purkinje cells, structures dependent on basal autophagy. An ATG4D variant previously has been associated with a recessive vacuolar storage disease in the Lagotto romagnolo. Clinical signs in the 3 Eurasier dogs also support abnormal function of the cerebellum, but disparities exist between the Lagotto romagnolo and the dogs in the study, potentially due to different effects of the P-Olive430THR variant in Lagotto romagnolo and the P-Olive375Al variant in Eurasier dogs. Additive effects of the MECR and ATG4D missense mutations cannot be excluded in dogs 1 and 2, which experienced a severe clinical course. The clinical presentation and imaging characteristics of the dogs were not consistent with the previously described cerebellar hypoplasia syndrome in the Eurasier breed caused by deficiency in the very low-density lipoprotein receptor, VLDLR. The study had some limitations, including the lack of clinical evaluation and investigations in dog 3, reliance on video footage and owner history, and lack of histopathologic diagnosis in any of the cases. Skin biopsies were not performed to assess dermal apocrine gland changes or establish fibroblast cell lines for mitochondrial activity evaluation. Additional work would include functional testing of these variants in an in vivo system, such as yeast complementation assays. The genetic analyses included 115 control Eurasier dogs not from the UK, which could have helped investigate transmission risks of MECR-associated PE, given the small breeding population in the UK. This study is the first description of PE with early onset of movement disorders associated with the MECR variant in dogs. The clinical manifestations in multiple littermates closely resemble those in children with MECR variants, strongly suggesting a genetic component, with MECR A to G as a potential candidate disease-causing variant. More marked clinical signs are present when dogs simultaneously carried mutant alleles at ATGD4 in a homozygous state, possibly suggesting additive effects. The results support further research elucidating the potential functional impact of MECR variants and their links to neurodegenerative disorders in dogs. Additional studies to investigate the frequency and distribution of PE-associated variants within the UK Eurasier population are warranted to promote responsible breeding strategies, given the large amount of inbreeding in this small population. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.